into the number of the chromosomes, we want to discuss how these chromosomes are arranged in the process of cell division. And in this case, what we see in the slide, it was referred to as meiosis. So when it comes to looking into the cell division, we have to understand that depending on the type of the cell, the specific process is considered. Specifically, there is what's called mitosis, and this is for the most cells in our human body, and it goes into through a set of phases. Eventually, it starts with 46 chromosomes and end up with 2 copies of these 46 chromosomes. Think about, for example, the brain cell, right? A new created brain cell should be exactly having the same copy of the other chromosomes. So the meiosis process is about cell division that will make a perfect copy coming from the other cell. Now, there might be some modifications in exceptional situations, but that is the aim in general. However, meiosis is a different type of cell division, and that is for gametes. So this is the type of division that is the cell is going to have in such a way that we'll have one set of chromosomes instead of 2. So meiosis reduces the number of chromosomes in a parent cell by half to produce 4 daughter cells. So if I show an example here, we can think of meiosis as starting with 46 chromosomes, similar to the case that we had here in mitosis. However, now we will go 23 separate and 23 separate here, okay? And then process is going to be completed through a second stage, okay? And then we'll have 4 daughter cells. Each would have the number of chromosomes of the parent. And this is essential for sexual reproduction in eukaryotes, like in humans, animals, plants, fungi, and so on and so forth. As you see here, meiosis involves 2 rounds of divisions, not only 1 round, 2 rounds of division. So that is why it's usually referred to meiosis round 1, and there's meiosis round 2. And that is why also the stages of, or the steps of these phases of meiosis, which are referred to as prophase, metaphase, anaphase, and telophase, which also take place in meiosis, would be labeled as, we see in this slide, as prophase 1, metaphase 1, anaphase 1, telophase 1, then cytokinase 1. And then we will go to 2 in the second round of production, ending up with 4 haploid cells. So that is meiosis that goes and takes place in a human body. And again, something to highlight here is that the primary purpose for this is to produce gametes. We're talking here about cells that make the sperm and the egg, okay? These are the types of cells there that is needed for production. And so the process that we are looking into is going to be building up the composition of cells, and these are haploid cells, as we discussed earlier, diploid and haploid cells. So something I want to clarify here as well, there are these different phases that meiosis goes through prophase, metaphase, anaphase, and telophase. I'm going to highlight some of the details there. But one of the things that we want to consider here is chromosomes, right? Chromosomes. Okay. And one of the questions that I want to ask you to note down here and also share as with me is, let's refresh with this. What is a chromosome? Okay, tell me, what are your own thoughts on what is a chromosome? I don't want you to search it online or find it in any resource. Just share with me the thoughts. How do you perceive or think about the chromosome? Okay, so we have now 46 chromosomes. Yet I'm going to sketch them in one specific way. Okay, let's say we have 4, 5 here. So these would be 10 chromosomes, and we keep going until we get 46 chromosomes. Okay, so 46 of these curves that I have drawn here. Through the process of this cycle of cell division, what's going to happen is that initially there are going to copies of these created. Okay. Oh, let me just switch the color just for the purpose of illustration. But I think let's keep it so that you don't see it as if, okay, we can use this color, green color here. Okay. So we will end up with another 46 curves here in this specific way, and we keep going. It's not the same karyotype that we saw before. We were looking at into 46 curves, and then another 46, which are showing sister copies of these chromosomes. This will end up with 46 chromosomes, and another term that I want to point out here, 46 chromosomes, but 92 chromatides. Okay, 92 chromatides, 46 chromosomes. Okay, so it is, in this case, here we have one chromatide, and this is another chromatide. So these two curves here, each one we can call one chromatide, which is a copy of the other one. Okay, so this might be a little bit confusing because you may start thinking directly about the karyotype that we have showed earlier, which is an illustration of the chromosomes in the human cell. But now what I'm showing here is, I'm not able to spend time to draw all the 46 here, but think about that I'm not drawing only 46 chromosomes. Think I'm drawing 92 chromosomes, but we don't call them 92 chromosomes because they are perfect copies. This is one chromosome. And now one would come and ask, how come it's one chromosome? Well, this is what we need to start imagining. This is now a cell division process. So there is a copying mechanism that already starts and takes place. And let me remind you about DNA replication, which is like what's referred to as interphase. Right at the beginning here, right at the beginning, all the informations are created. Okay, DNA replication already takes place here in this stage, and so all the genetic information you see here, we have one chromosome and another set of chromosome. All these chromosomes, it is a diploid cell. That is how meiosis starts, with a diploid cell, and then all the information is duplicated. Later on, there are 4 phases that are going to take place. So it's there is a prophase. So in this case, what we are looking into here is, I'm going to highlight a little bit more details in this prophase, because there is a very important process, especially that takes place in meiosis, that's called crossover. And this is the key. If one would understand that why babies are having slight variations, and we're talking here about haploid cells coming from the sperm and the egg, how come there are going to be variations? It is this where this process takes a place as part of the prophase, which is called crossing over. Okay, crossing over. So look into this in the shortly. Prophase, that is the first stage here. You can see the chromosomes being arranged in this way after they are duplicated. So they start taking copies of these chromosomes. Next, in the metaphase, they are going to be set in the middle, somewhere in the middle. The chromosomes are distributed here, and then next in anaphase, they are going to set towards the end side. So it is kind of pulling the chromosomes needed, because eventually this is going to be into two set of cells, and then the division would take place. Okay, so another round will take a place. Now we don't see crossover here in the second prophase, and as it would happen or take place in the first type of the prophase. So there are certainly more in-depth details into the meiosis and all of these specific steps. Let's say metaphase, again, is about pairing, and anaphase is about separating, and telophase is about producing the two cells. So maybe that is a way to see this. This is mating, so things start getting in the middle. Anaphase is about separating the homologous chromosomes, these chromosomes that look at exactly like each other. Great. So as I mentioned earlier, one of the important processes that takes place in the stage of the prophase, it's what's called crossover. And crossover is, let me try to draw something here. And it is for transferring genetic information, and it's a very interesting process. So if I draw here, so we have one chromosome here, two chromatides in this case, and then have another chromosome. We are going to call these two as homologous because they have the same size, and they're going to get close to each other as the prophase process, which is the first step in the meiosis. And these homologous chromosomes are going to align more in the metaphase stage. So anyway, they start, we're looking to one gene, for example, here, one specific gene. Actually, let me use a different color here for this gene. So as these two chromosomes come close to each other, there is a process that's called crossover that may take a place for in the meiosis to eventually achieve certain variations. So we can think of like taking this area and cutting it from here and replacing it with the green, and maybe doing the same thing here and replacing this with the other color. So let me take this and then replace this with blue. Okay, this is what I'm going to do. And then also replace this with blue. This is also another thing we can do. And then we can actually go and color this with green. Okay, and this is kind of a crossover operation. If I were to give ACG as an example, TTAT, and this is gene, right? And it is the same gene that we're going to look into. Okay, it is the same gene. Okay, and then let's say we're crossing over while we keep this side here. Okay, sorry for this. Let's use actually a different gene. Okay, it is a different gene, and then what's going to happen as a result of the crossover for the purpose of, oops, sorry, I missed. Captions went away. Yeah, it's back again. Okay, sorry for that. So let me use a different color here. Sorry for the confusion. If we had just exactly the same gene, we're not going to get any product or a different outcome in terms of crossover. So we're looking here into ACAD. I'm referring to this, for example. It could be even different, okay? We can have AG here. And then the crossover, in this case, AC and AC is going to lead to, in terms of crossover, is going to stay as AC. And this can be changed and resulted in two outputs, okay? One, which is going to be GAA, and we take this one here, which is going to be AT. So this is one new level of information, okay, as a result of crossing over this part with this part here and keeping these other parts. So we have crossed over this with this, and then we get AC, GAA, AT, okay? So this is one of the examples that shows how crossover takes place. It is kind of taking two sequences, and then there is a specific area that is going to cross over to the next one, and then that is, you can think also, another variation would be is that we keep AC here in the second one, and then we have G, T, T, and then that was AG, okay? So the second sequence is as is, except the 3 alphabets in the middle, and the same thing for the first one. So just an example to illustrate how we might go through crossover in this process, and what we can refer into as well as recombination. So here's an example in this slide as well, example illustration. These are the parental DNA molecules. So we have coming from two parents, and then as this process comes into the intermediate phase, there is kind of the chromosomes are coming to each other. This is coming from mom. This comes from dad. They are homologous. They have the same size, so they're gonna come close to each other, and somehow they will start folding. As they fold in this stage here, and as they meet each other, we're going to end up with this recombination. And this is similar to the example that I was trying to give. What you see here is this area, okay? Well, this area here has crossed over to this part, okay? And this part has crossed over to here, okay? In this case, as I gave you in the example, the middle part. Okay, so let me give you an example here and see if you can get it well. Hoping I don't confuse you. So let's say I want to cross over this sequence ATT. Okay, so these are two sequences. And let's say I'm going to I want to cross over the last 3 nucleotides. What's going to be the output? Okay, only the last 3 nucleotides. What's going to be the output? So share it with me as you give me the answer. So crossover is happening during fertilization, and this is what leads into the genetic diversity that we see as part of the cell division. And this is the beginning of the chromosome crossing over. As I said, we have here the homologous being aligned together. Eventually, this coloring indicates the replacement of these parts, okay? And this, each time there is the sperm coming and egg coming, the outcome could be different, right? The outcome based on this process could be different. Now, an interesting question would be for somebody is asking maybe this question is, we see now that the process of crossover is taking a place in the cell, right? And we see this for the purposes of the reproduction. Now, one example or one question one might be interested with is that happening in through mitosis, the other cell division as well, or how does it go? Okay, so in general, in mitosis, as I mentioned before, mitosis is this cell division that wants to produce two daughter cells, and they should have the same number of chromosomes as the parent cell, because the aims are primarily for growth, repair, and maintenance. So, although crossovers are clear step in or part of the meiosis, they are rare events in the mitosis. Okay, they are generally uncommon and may lead to abnormalities in the case of mitosis. So thinking that crossover could happen like this, and this could be the output. Imagine this was brain cell, and this was an eye cell, okay, for example. Okay, so crossing this over could cause some big issues. So ploidy, as it refers to the number of chromosomes attached to it, a certain range or spectrum of levels that we can look into here. Okay, so we looked earlier into haploids, can called monoploidy. There's diploids, diploidy, 2N. So these are part of what's called euploidy. Okay, so in general, haploid were the cells with one set of chromosomes, human sperm and egg cells. Diploids, the cells with two sets of chromosomes. Okay, we already discussed this earlier. Polyploidy generalized this. Okay, polyploidy generalize this into saying that these are cells that have more than two sets of chromosomes. Okay, it could happen in some plants, like for example, large fruits or so. There is also, and these are all part of euploidy, which means having normal and balanced number of chromosomes. Okay, so this is what we may think of more of the normal case, because there are balanced, there are a balanced number of chromosomes. There's aneuploidy. It is a condition where the cell has an abnormal number of chromosomes. In this case, maybe that is by missing some chromosomes or even having extra. Okay, missing or extra. Now this can be awkward in terms of the normal view that we are having, but one of the common things that or diseases that sometimes happens are attributed to trisomy. Okay, and trisomy, it is a type of this aneuploidy where there's an extra one chromosome. So, and specifically, it is the case where instead of having two copies of that chromosome, there are going to be two copies, one coming from dad, one from mom, and then an extra one. Somehow that came into play, okay, instead of the usual pair that is there. So this is a trisomy case that I referred to as part of diseases, like one of the very known diseases with an issue with the number of the chromosomes, the Down syndrome, where chromosome 21 is having 3 copies instead of 1. And Down syndrome, it is the most well-known trisomy. It causes, as some of you may be aware, it is this kind of disease that may lead to intellectual disability, some facial features, and so on and so forth. There's another kind of syndrome where here, again, chromosome 18 is going to be having 3 copies. And this causes development delays. And there is Patou syndrome, where chromosome 13, if we refer to the karyotype, chromosome 13 is going to have 3 copies instead of, again, 2. And this also leads to several intellectual disability and physical abnormalities. These trisomies results from errors during the division of parent cells, and somehow end up with having an extra chromosome being present in the egg or the sperm. Now, most of the types of the trisomies result in miscarriage, okay? So somehow the process is going to end up discarding these cells, and there might be at specific stage of the embryo and so forth, but that's going to be kind of a miscarriage. These can result, as you know, in issues with the births. However, still, some are going to stay and then lead to, as we said, newborn babies with some of the issues like these diseases, and the reason is not fully understood why. Now, one of the things that we want to also arrange in our view of the chromosomes as we start aligning things together and put our minds around, and in the context of the evolution concepts, which is this mechanism of process where we are able to spot what? We're able to see some genes, as you see here, across different species, okay? So some genes are part of different species, in this case, but they are the same reference gene, and that's when we call them orthologs, okay? Orthologs in this case. Paralogs in the in other case, it is thought to or viewed to be the same. We are having one gene here, and it is some kind of mutation or some changes that gonna still be the same type of gene with some change, okay? And that's what the paralog. So in other words, this hierarchy here actually is useful to summarize the concepts of orthologs, paralogs, and homologs. Homologs, again, we are trying to refer to some sort of similarity between the different chromosomes, okay? So what I want to highlight here, I just want to look something into I missed from my notes, so bear with me. Yeah, I guess I had something to add. Anyway, so this is the gene duplication process. We see here a group of species. This is one reference gene. Some modifications happens. Eventually, we can reference all of these species, giving one type of gene that they all share that gene. And so that's an ortholog, okay? Paralog is going to be gene. We're going with the gene, but with some changes within the same species, okay? And so this is very useful as we want to look into the set of genes, and we want to characterize them with our studies of the different species. is very helpful and informative to understand the genetic similarities across the different types of species. For example, one of the typical analysis that I've came through in my research earlier or looking at other projects when it comes into the study of orthologs and paralogs. What if I have now a new species? Okay, this is a new species. It's not a well-characterized species. Okay, and then I discover a new gene. Okay, there is this some sort of gene. Sorry, this is like TAC. Okay, something. Okay, some gene that we have here. I'm sorry, some latency here is not showing the font clearly. But anyway, so there is this gene. I'm not sure of the function of that gene. So I go to the other species that are model species and try to match this with the specific gene. Now, I may be able to have an exact match. I might not be able to have an exact match. And through going at the different layers of matches, maybe eventually I'm able to find an ortholog coming from another species that will help me characterize the function of this specific gene. So here's one interesting example and one of the ways to align the sequences. A question for you here: what type of sequence that you are seeing here? So share that with me as well. I'm going to give you a moment here. What type of sequence do you see here in this over this slide? Great. So this is a list of amino acid sequences for the proteins, and you can see there is between human, chimp, mouse, and rat, there is certain conservative regions coming from this histone H1 case, and there are some regions that are non-conservative. Okay, so they are not exactly the same amino acids across all. Sometimes it is semi-conservative. Okay, so it is preserved in certain areas, right? And sometimes, as we see here, it is non-conservative. Okay, so depending on that kind of amino acids and the reflection of that change. So this alignment that you see here, which we refer to aligned sequences, can help us to see the different parts of the conservative regions and try to tell us what parts are orthologs of those genes and so on and so forth. So now I'm going to refer to another important concept as we have considered looking into cell division, chromosomes, and one of the things that can be attributed in terms of these biological processes, for example, crossover and the changes that it may lead to, and that if there are issues in the cell division, we may see some diseases. Mutations come to be also one of the changes that may happen to the sequence of DNA. This could be because of several reasons. Okay, so DNA here. DNA may be exposed to certain mutations or certain changes. Okay, for example, during the DNA replication process. We already saw this when there's a forking and then there is the Okazaki fragments. We have talked that there is going to be a repairing mechanism. And part of that is what's called deamination. We will see some more information about how the DNA is repaired and shortly as well. But anyway, during DNA replication could be one of the cases where there might be some mutations or maybe exposure, for example, to radiation. Okay. So exposure to radiation or chemicals could have such impact. There are different types of mutations. There's deletions, for example. Okay. Deletions involves removal of one or more of the nucleotides. Okay. So imagine we have this sequence ATGCA, for example, And deletion might result, if we're going to delete G, okay, result with a shorter, with this shorter sequence, okay? If that deletion is going to impact an amino acid, because we know now each tree would code for a codon if they're part of, again, coding genes, and eventually this is going to be part of some codons, right? And again, as an example here, that may affect the production of that protein. There's going to be specific types of mutations, like, for example, missense mutation that we're going to also talk about. But in general, we have deletion. We may think about insertion as well as one type of the mutations. So imagine we have, again, A, T, G, C, A. And what if A is added towards the end? So an extra A is added here. That is also kind of one mutation. Or there is what's called inversion, where one part of the DNA is reversed, okay, inversion. And so this is also another kind of, let me put this example again. So this is another kind of mutation. So in this sequence, an inversion might flip a certain part of it, okay? So this might flip here and becomes CGT, okay? T is here, C is here, and G stays here because flipping it. And this is what's called inversion mutation, okay? And there are also other types of mutations like substitution, duplication, or frame shift, which is which is caused by insertion or deletion that are not as multiple of 3 nucleotides. So let's look into mutations. As I've discussed already, there is deletion. We're taking some part here of the chromosome, ending with the shorter chromosome. Duplication, this may lead into expansion of that chromosome. Inversion is going to be flipped, as I gave you in the example. Insertion, and so on and so forth. So these types of mutations may really impact the outcome, and not all mutations are necessarily linked to diseases. So, for example, sometimes we are able to characterize certain mutations to be linked to one of the diseases, okay? So imagine one cancer patient, breast cancer patient, okay? And then, for example, one of the genetic testings that are, or the genetic testing done at certain stage, is to look for BRCA1 and breast cancer, BRCA1, BRCA2 breast cancer genes, and see if there are any recognized mutations. Now, the patient could be having breast cancer, and there are no mutations in any of those, okay? And they might have some mutations that are linked and characterized to be a part of the group that have been receiving this disease. So if you have a normal population and a population with a disease, this is a normal population. We're building up the discussion into the experimental design that we're going to come later on. And then there's this disease group, okay? Disease group, normal or control group, whatever. So we have two groups, and the disease group generally is having certain mutations that are linked to be causing breast cancer, or maybe not causing, let's avoid this term for the time being, but linked to cancer disease. But sometimes there might be mutations that are unrecognized. In that case, we know the population of those who are normal and those who are having disease, but we are not able to recognize this. Maybe we'll discover it in the future, and so on and so forth. So part of the genetic testing report that is shared with the patients is to tell them, for a list of maybe roughly 18 genes or so, if they had any positive mutations or negative or unrecognized mutations. So sometimes mutations, not every single mutation is going to mean specifically or necessarily a disease in the process. But here's a list of examples that you can see in this picture, giving or highlighting some of the diseases, okay? Like, for example, there's sickle disease here. Now, what you see in this depiction, it is interesting. 1. So this is the first base here. This is the first base, right? And looking into mRNA in this case. Then the second base, this is the second base, and each row gives us the complete, when we shift from one row to another, we're going to see, oh, sorry, it's from one row here to another row, we will see the different third bases, okay? So these arrows here indicates if there were some types of mutations, so there is insertion, deletion, like in this case, this is Huntington disease, it's kind of insertion, okay, that takes place in this area, okay? Now, there is sickle disease, for example. In this case, it is some missense mutation, okay, that's going to change glutamic acid to valine, for example. So this change that's going to reflect onto the amino acids and the codons that makes the proteins are going to, in certain cases, be linked to some types of the diseases. Sickle disease is one of these classic examples caused by missense mutation. So instead of having the glutamic acids, lysine, the protein or the hemoglobin protein is going to have valine lysine, which is going to cause, I mean, it is minor. It might be perceived as a minor change. It's going to have significant consequences. The altered hemoglobin is going to be called hemoglobin S and tends to cluster together, and eventually this will cause red blood cells to deform into what's called sickle shape. Okay, now this is also here illustrating another type of mutations. So what's called nonsense mutation. So let me introduce this to you a little bit more in more details. So nonsense mutation is a type of mutation where there's a creation and a change in the DNA sequence and going to lead to creation of premature stop codon. Okay, stop codon. This would really affect and may halt the process of synthesizing a protein. So imagine that there's going to be a mutation that's in the DNA that can impact the process of creating a specific protein. So let me share an example here. Let's say we have ATG, GCA, and then CCA, and then TAA, okay? Let's assume, oh, sorry. Let's say we have this example here, okay? ATG here that you see is the start codon, signaling the start of protein building process. And TAA is the stop codon, okay? This is the stop codon here, which is going to say or send a message or stop here, stop building the stop building the protein, okay? Now imagine a mutation changes the second codon, this GCA, okay? Let me change the color here to red, okay? In this case, imagine this changes to imagine this changes to the stop codon, okay? And becomes TAA, okay, in this case. If this kind of mutation is going to happen, it's really going to cause an issue because the protein synthesis is going to stop just at the next amino acid, okay? So here we had GCA, and we have looked into changing it, and you know, by GCA specifies alanine, okay, and if we replace it by this stop codon, we will lose the process of creating that alanine. So what we see here is this arrangement of OPNY1LW. So it is this gene that we see here, and it's arranged. There's one LW, one MW1, one MW2. So it's kind of head to tail arrangement. Okay, and this is the normal color vision. So imagine that this is linked to our human eye. And if we have this arrangement for this gene, which is this one is linked to red color or red cones in our eyes, okay, and those are part of green cones. And the cones, if you know the anatomy of the eye, we have the cornea here, right? We have the lens here, and then we have the retina here. And part of this area, there are rods and rods and cones. Okay, and cones are responsible for capturing the color, the electromagnetic spectrum that comes to the eye, and capturing the different levels and layers or levels of colors. So if anything happens with the cones, we are gonna lose something with our vision. So a deletion may end up that we don't see. It's green color blind. Okay, if there's some stop kind of mutation like the missense type of mutation, is going to certainly impact having this gene and eventually the protein linked to it created properly, and so there would be issues with the cones, so they are absent, and so this is a red color blindness. And then you can look into these different variations. So overall, these changes or mutations may impact the functionality in the subject. Mutations can come into different types. There's missense mutation. Sometimes can be acceptable and unacceptable. Nonsense mutations, we looked into deletions, insertions, and these are some of the examples that they may cause. Okay, there is this frame shift mutation, and again in this frame shift, it is kind of insertion or deletion, and are not in multiple of 3 nucleotides. Okay. So let me show you an example here for frame shift deletion. So let me put this as a sequence here, ATGCA. So this is the sequence. If we're going to delete the T, for example, we will end up with AGCA. Now, what is this going to cause? It's going to shift the reading frame of all subsequent nucleotides. Okay, all subsequent nucleotides. So in this case, we had ATG, and then next one CA, and continue. Now, the fact that this is shifted, the coder now is going to start with AGC. Okay, so this is a shifting on whole of the translation process. Okay. So one of the exercises here that I want you to do is note this question down and tell me, although I did not explain it, what is meant by a silent mutation. So what I want you to do is first write it down. What could you imagine from the name? What could be a silent mutation? Okay. And also search online and see if you had gained any new insight about silent mutations. So I'm not going to explain it for you here. I would expect it to be answered in the exercise list that you have for this lecture. So I'm going to stop here for now, and then we'll continue discussion on DNA repair mechanisms on the coming part.